Hi, everybody, and welcome to our module on blotting. Blotting refers to a number of laboratory techniques designed to identify a number of different types of molecules. There is the southern blot, which identifies DNA in samples. There is the northern blot, which identifies RNA in samples. And the western blot identifies protein in samples. So we'll talk about all of these laboratory techniques in this video now. The southern blot is a lab technique named for its inventor, Edward Southern, and the other types of blotting, like northern and western blotting, were named to distinguish them from the name southern based on the directions on a compass. And a southern blot uses a probe to identify the presence of DNA in a sample. So let's talk about what a probe is. A probe is a single stranded DNA molecule that carries radioactive or chemical markers, and it will bind to complementary DNA sequences. So the probe is sometimes called cDNA because it's complementary to the DNA you are trying to identify. And when the probe binds to the DNA in your sample that you want to identify, this is called hybridization. And once the probe has bound, the markers in the probe reveal the DNA in the sample. So this is a picture just to help you understand at the bottom. We've got a probe, and we want to know whether our sample contains the complementary base pairs to that probe. So we stick the probe in with the sample, and if it binds, then the probe can be identified by its markers, and if it binds, then we know the complementary sequence must be present in our sample. The way a southern blot is performed is by taking a DNA sample which contains lots of clumped up DNA and then mixing it with substances called restriction nucleases, which are enzymatically active and can cleave the DNA into lots of little tiny pieces. The pieces are then placed on a gel electrophoresis, which uses electricity to separate the pieces by size. So, what you will get is a spectrum of all the pieces of DNA separated based on size. This is a picture of what gel electrophoresis looks like. It's called a gel because it has a gelatinous material here. The DNA sample is put at the top, and then electrical charges are applied to the gel, and the DNA will separate based on size. The ability of the pieces to move further along the gel is based on how big or small they are. So once the pieces are all separated by size, you then perform the blotting. This means that you put a piece of filter paper over the top of the DNA pieces, and the DNA will stick to the filter paper, which you can then use to do further testing on. At this point, you add the probe and you wash away any unbound probe, and only bound probe will remain. The filter paper is then exposed to a film, and the bound DNA in the probe is revealed. So, for example, even though there might have been pieces of DNA separated by size all along this gel, the probe only bound to this piece here and this piece here, indicating that those contained the complementary DNA to that probe. And this is often done with multiple samples. So, for example, you can see that in sample 1, the DNA probe bound to two pieces of these two sizes here. In sample 2, the DNA probe bound to pieces of other sizes. Here's a summary slide of how the southern blot works. You apply your DNA to sample wells, you use electrophoresis to separate the samples based on the size of the pieces of DNA, and then you apply a probe, and the probe will identify which pieces of DNA in the sample contain the complementary bases to the probe. There are many uses of the southern blot technique, but I'll go through the two on this slide, which I think are worth knowing for your boards. Restriction fragment length polymorphism analysis is a technique used to characterize genes. This technique involves using enzymes called restriction nucleases. These are the same enzymes we talked about earlier when discussing southern blotting. These are DNA cutting enzymes, and these enzymes will cut DNA every time they encounter a specific base sequence. For example, they might cut DNA every time they encounter a GTGCAC sequence. When they do this, they create fragments of genes, and RFLP analysis involves analyzing the fragments that are created from these restriction nuclease enzymes. It turns out that different genes break up into different lengths of fragments, and therefore the lengths of the fragments created by these enzymes is unique to each particular gene. You can then use southern blotting to detect the lengths after fragmentation. To understand this, look at the bottom of the screen. Imagine that we subject gene A to a particular restriction nuclease enzyme, and it breaks up into two pieces, one that is 1 kilobase in size and another that is 1.5 kilobases in size. We then subject gene B to the same nuclease, and it breaks down into only a single fragment length of 1.3 kilobases in size. So it turns out that these fragments are unique to each gene, and we can use them to characterize the gene. When you think about it, this is very clever. It would be very cumbersome to identify all the nucleic acids in a gene, but you don't have to. You can just break the gene up into fragments, and the fragments will tell you something about the gene you are studying. So you can imagine then that we could analyze an unknown gene and see what it breaks up into. If it breaks up into fragments just like gene B, we would know it's similar to gene B. If it breaks up into fragments like gene A, we would know it's similar to gene A. And here's an example from the web just to show you how you can use restriction fragment length polymorphisms to determine the genotype of a patient. So in this example, patients who are AA, meaning they have two copies of the capital A gene, create one fragment of this size. Patients who are little a create one fragment of this size, and patients who are heterozygous create two different fragments. We can then go ahead and analyze a family and determine which patients are heterozygous carriers, which patients have two little a's, which patients have two big A's, and so on, all by using restriction fragment length polymorphisms and the Southern blot technique. Sickle cell anemia is another situation where Southern blotting can be used. A normal beta globin gene breaks up into two fragments, one that is 1.15 kilobases in size and another that is very tiny, 0.2 kilobases. A sickle cell beta globin gene breaks up into a single fragment that is 1.35 kilobases in size. So this fragment will only be seen in individuals who carry the HBS gene. What this means is that presence of a 1.15 kilobase fragment identifies a normal beta globin gene, and presence of a 1.35 kilobase fragment identifies a sickle cell beta globin gene. So if you look at the bottom of the screen, after Southern blotting, a normal individual will have only one band in the 1.15 kilobase range. On the right side, in an individual with two sickle cell genes, you will see only one band in the 1.35 kilobase range. And in a carrier, you will see a mixture. You will see some fragments from the normal gene and some fragments from the sickle cell gene. And if you understand a southern blot, it's very easy to understand a northern blot. It's the same technique, except that it's used to identify RNA instead of DNA. So once again, you separate the RNA pieces by size using electrophoresis. You then apply a probe that is complementary to the RNA that you're looking for to determine if it's present in your sample. And this is useful for studying gene expression because if a gene is being expressed, it should generate mRNA. So if you find the mRNA that corresponds to a particular gene in your sample, it suggests that gene is being expressed. And then if you understand southern and northern blots, it's easy to understand western blots. They are the same technique, except we do this to identify protein in a sample. So once again, you separate protein molecules by size using an electrophoresis. And then instead of a probe, you use a specific antibody to the protein that you're looking for. And then you use blotting to see if the antibody bound to its specific protein. And Western blotting has historically been used many times to detect antibodies. It can look for IgG or IgM in Lyme disease or IgG of HIV-1. In some cases, this has been replaced by other techniques, but in some cases, it's still used. The last technique I'll mention is a southwestern blot. This is used to study DNA protein interactions. It gets its name because it combines features of a southern and a western blot. In this technique, proteins in a sample are separated by electrophoresis, just like in a Western blot. However, instead of adding an antibody like you would in a Western blot, a DNA probe is added similar to what you do in a Southern blot. And therefore, this is used to study DNA binding proteins. It's especially useful for looking at transcription factors where you want to know if proteins bind to DNA. And here's a picture to help you better understand how this works. You add a sample looking for proteins. You separate the proteins using electrophoresis by size. You then add a DNA probe and you see if the probe binds to the proteins. If it does, it suggests an interaction between this particular strand of DNA and the proteins in the gel. And that concludes our module on blotting techniques.